So this presentation is going to be more about the who, what, when, where, why, and how of neurotransmitters. So we talked about synapses before. Now we're going to talk about the neurotransmitters, what some of their roles are in that. Um, so a couple quick things. We have some different categories of neurotransmitters. We have amino acids, small organic molecules. So some of these are like glutamate, glycine, GABA, which is gamma aminobutyric acid, short for that. We have amines, which are small organic molecules, which are going to be slightly larger in size. Those are some neurotransmitters like dopamine, acetylcholine, and histamine. And then you have peptides, which are short amino acid chains, aka proteins, which are stored and released from secretory granules instead of the standard synaptic vesicles, because they're a little bit bigger. So the dynorphin and enkephalins, substance P is one of those. So some examples of their sizes and shapes you can see here. In A, we have the amino acids. We have glutamate, GABA, and glycine. Right? Then we have our small molecules, acetylcholine and norepinephrine. And then we have substance P, which is our small protein. So we see we have a couple different, like three major categories of neurotransmitters. And here's kind of some of their structures. So synthesis, okay, especially for some of the things that are going to be in secretory granules, those are going to be actually packaged all the way up here, okay, up at the ERs where they're going to be synthesized and then packaged and then transported all the way down the synaptic terminal. The synaptic vesicles can go through more of a localized synthesis depending on what the neurotransmitter is, where they can actually be synthesized in this area and then packaged into these vesicles down here without having to make its way back up to the soma. Okay, so now we've got our neurotransmitters. They're packaged in their vesicles. We need to get them to bind. We need to get them to be released. So where vesicles are sitting at, and some of the vesicles are loaded into the active zone we talked about in the last thing. I'm going to see it's kind of docked and ready to go right here. Then all of a sudden, action potential comes down, okay, causes calcium to rush into the cell, and that's going to interact with these proteins down here that are part of the kind of what's called the snare mechanism, the things that are capturing the vesicles and loading them at the docking location. When those get activated, they cause this membrane of the vesicle to fuse its way into the membrane of the cell. And when that happens, it kind of opens up the vesicle and neurotransmitter spills out. Then the membrane gets pinched off again and the vesicle floats away. And now we have a vesicle that's missing some of its neurotransmitter that's moved away from the active zone. And it's ready to be refilled with more neurotransmitter. So now that we've got our neurotransmitter out in the synaptic cleft, what happens? Well, we have two major types of receptors, two major categories of receptors. They're the ionotropic and metabotropic receptors. So first off, the ionotropic receptor, that is when a neurotransmitter comes and binds the receptor, causes a transfer, so a neurotransmitter might come in here. In this case, it's an acetylcholine receptor. It's going to bind to that receptor and cause it to open a channel. This allows for fast direct signaling. So if we look here, we have nicotinic acetylcholine receptors expressed in some tissue here, and they apply 300 micrometer acetylcholine. You can see the quick response here. And the black is like without any of the drug that they're applying. It doesn't really matter what drug they're talking about. Just give an example of how quick that current is when the acetylcholine gets thrown onto the nicotinic acetylcholine receptors. Okay? So they're fast ionotropic, meaning that they open up and ions just start to flow through. Okay? Then we also have the metabotropic receptors. These are ones where a neurotransmitter binds the receptor and activates some kind of G protein coupled cascade. Okay, it could be a second messenger cascade that gets activated, or that G protein might directly activate some ion channels. So you see we have our two options here. So you have the neurotransmitter comes in and binds, activates the G protein, and that G protein activates an ion channel. So it's a G protein gated ion channel. Or we could have that neurotransmitter come in, bind, activate the G protein, which in turn activates an enzyme which might activate a whole bunch of secondary messengers, which may modulate the neuroactivity or might end up opening up more channels. A lot of times, but in this case, so it's going to be some kind of neuromodulator that's going on. I mean, it's going to kind of not create a signal in the cell, but modulate and change the signal that's coming in from some other neurotransmitter. So as an example, muscarine is the agonist for the acetylcholine receptor that's metabotropic. So you can see here in control with no muscarine, they get this signal. When they apply these current steps to the cell, they get incoming current. And when they apply muscarine, all of a sudden it's been reduced. So in muscarinic or metabotropic acetylcholine receptors, you actually get less current coming in. So it's actually an inhibitory thing, as opposed to in the ionotropic, which were very extremely excitatory. Okay, so great. Now that we've got the neurotransmitter in there, we got to get rid of it. Okay, it can't just stay in there forever. If it stays in there for too long, we're not going to be able to send another signal to that cell. So what are some things we can do? Well, one, we could just rely for diffusion. We could just rely for that neurotransmitter just make its way out and disappear out of the synaptic cleft. Second two options are reuptake. Third options are reuptake. One is where it's retaken up by the presynaptic terminal. So in the presynaptic terminal, okay, you might over here have some kind of reuptake channel. And as that neurotransmitter makes its way out of the synaptic cleft, it'll get sucked back into that presynaptic cell. Also, that neurotransmitter might get pulled into nearby glial cells. So we have that synaptic terminal, okay, and it's going to have glial cells around it. This may be an astrocyte right here, okay, the edge of an astrocyte. And as that neurotransmitter kind of starts to diffuse out, it might get pulled into that glial cell, and that glial cell can recycle it and return it to the neuron to repackage into a new vesicle. We can also have breakdown via enzymatic destruction. So some kind of esterase that's going to go in there and break that neurotransmitter down. And then we can also have desensitization, meaning that the neurotransmitter just no longer affects the receptors. They just stop opening due to it because it's just been around for too long and they're no longer responding. So we have those kind of 5 options that we can have to get rid of neurotransmitter from the synaptic cleft. Great. So a couple other things about neurotransmitters. We want to talk about neuropharmacology. So neuropharmacology is extremely important because we use drugs all the time to block channels or open channels or modulate channels. We use them. We take drugs that do that help us, but scientists also use them to study things about receptors and what their activity is. So the first thing might be somebody might use an antagonist. An antagonist is one that's an inhibitor of neurotransmitter receptors. So an example of that is this curare. Curare blocks the binding of acetylcholine at the neuromuscular junction. So you can see this guy up here with his blow dart. Curare actually comes from a plant that's found in the rainforest, and you mash it up really good. You put it on the tip of a blow dart, you shoot something with it, and you've paralyzed it. So it's a great neuroantagonist that blocks the binding of acetylcholine, so it basically stops muscles from contracting. Okay? You can also have agonists. They're drugs that mimic kind of the effect of the naturally occurring neurotransmitter. So, for instance, one of those is nicotine. Talked about a little bit earlier with the ionotropic acetylcholine receptor. Okay, normally acetylcholine comes on and you get a nice quick response like this. Okay, this might be with acetylcholine. Okay, but you put nicotine on there and you're going to get the exact same response in the ionotropic receptor. So it's good to understand we have these two kind of roles that a drug can play. We have things that block effect, and we have things that mimic the effect of different neurotransmitters. Okay, so what are they blocking? What are they allowing in? Okay, these are things called receptor potentials. So we have both excitatory and inhibitory postsynaptic potentials, also known as an EPSP and an IPSP. EPSPs are quick postsynaptic membrane depolarizations, okay, meaning we're going to raise the membrane potential up based on the release of a neurotransmitter. Okay. IPSP is an inhibitory postsynaptic potential, meaning that neurotransmitter binds and it causes a decrease in the resting membrane potential. So when you think about excitatory is going to bring it up, and inhibitory is going to bring the membrane potential back down. So they're contradictory effects, and they're only based on location. Sometimes neurotransmitters will be excitatory, sometimes they'll be inhibitory. Depends on what location in the brain they're in and what the postsynaptic receptors are. That plays a big role. So kind of, if we think about this a little bit, what the cell does is it does a lot of synaptic integration. So say we get one action potential, great. We get this small EPSP when we record it in the soma, right, in this first one right here. We get one action potential, we get a small EPSP. However, now we get 3 action potentials that come in, okay, and we spatially, they'll add together as they're making their way this way towards the soma, and now you can see we've got a much larger excitatory postsynaptic potential, basically increasing the chance. This is increasing the chance of reaching threshold, right? So more likely to produce an action potential. Again, EPSPs and IPSPs are not action potentials. They are changes in the membrane potential, which could potentially lead to an action potential. We also could have temporal summation, right? You have one axon coming in, and also we have 3 signals there, and those 3 are going to overlay in the soma, so they're going to make their way down here, okay, and kind of overlay in the soma. So the first one raises it there, second one raises it a little bit more, third one raises it up here. Okay, if the timing's fast enough, we might reach threshold again. If the timing's too slow, right, then we might not hit threshold because, well, those voltage-gated calcium-potassium channels might turn on and drive that membrane potential back down. So the time frame is really important with temporal summation and with spatial summation to make sure we're getting high enough depolarization and also a fast enough depolarization to get an output signal. Okay, cool. So now we've got all those output signals. We've got all these signals coming in from all these different places. We're generating excitatory postsynaptic potentials. Well, what's happening with the inhibitory stuff? Well, it's going to work as what's called a shunt. So shunting inhibition. So here we get an EPSP coming in, right? So we have our excitatory synapse that's active. Sends that signal down here. This inhibitory synapse is inactive. So it just keeps making its way down to the membrane. And then we look in the soma, the signal is still there. It's not quite as strong as it used to be because we're going to have some degradation as we travel along the axis, as we travel on the dendrite, but we do have a depolarization, right? And if we had more from other cells, they would hit that and they would lead our soma to try and hit threshold, right? However, if we have inhibition, okay, that excitatory postsynaptic potentials make its way down, coming down the cell, and all of a sudden we get here. And an IPSP is going to drive that membrane potential back down, right? We're allowing some kind of inhibitory current, meaning the 2 types of inhibitory current are typically chloride going in, or we're going to open up potassium channels and let them come out more, okay? So this would be in or potassium coming out. Okay? Those would be the 2 things that might drive down the membrane potential. Typically, it's going to be this one, the chloride coming in. But by driving that back down, we've basically negated the effect of that EPSP, and now we have 0 change in the membrane potential of the soma, okay, because we've shunted that excitation, kind of blocked it by adding inhibition after the excitation. So a way of regulating kind of like which neurons get active based on which inhibitory interneurons are active too, trying to block some of that activity. Okay, so basically that's it. There's not too much we'll talk more. We'll get into more details as you dive into your neurotransmitter project. You pull out some of those different neurotransmitters and explore what they're about. But basically, we've got a rich diversity of chemical synapses to allow for lots of different types of behavior. Okay, there's so many neurotransmitters out there, and you'll see the list. And it's not even a full list. And not even just the number of neurotransmitters, but also the number of receptors that they each have. So there's going to be lots of explanations for drug effects here, and explain all of that. Why when people take drugs for depression, they take stimulating drugs, effects of caffeine. All of these are going to have different receptors that they activate and act on, and they're all going to play different roles in the brain. And other things with synapses, defective synaptic transmission, is the basis for many neurological and psychiatric disorders. Seizures are excessive excitation, right? You have too much serotonin in your body that's going to lead to the inability to feel happy because you can't get rid of serotonin, and you become desensitized to it. So there's all sorts of different things going on. And it's also going to be key to understanding the neural basis of learning and memory, because, well, receptors are all about creating synapses and creating that signal, talking from one cell to another.